I'd like to start off by thanking REPROCELL for organizing this webinar today. We've had a great experience working with their STEMGen iPSCs for the development of our atrial cardiomyocyte product. As Bob mentioned, my name is Anne Sheldrake, and I'm the principal scientist here at STEMbiosis. Joining me today will be Dr. Todd Herron, our collaborator at the University of Michigan and director of the Cardiovascular Regeneration Core Laboratory there. He's done some incredible work using SelvoMatrix Plus to mature iPSC-derived cardiomyocytes, as well as to characterize STEMbiosis's chamber-specific atrial cardiomyocytes, and that's mainly what he'll be presenting on today. I'm going to be starting out by giving a brief introduction to STEMbiosis's core technology, our cell-derived extracellular matrices, which we sell under the trade name SelvoMatrix. I'll describe the rationale for the use of cell-derived extracellular matrices and how we arrived at using them to support the maturation of iPSC-derived cardiomyocytes. And then I'll turn it over to Dr. Herron to discuss his results on the chamber-specific atrial cardiomyocytes. And as Bob mentioned, we'll also leave time for a Q&A session at the conclusion of the presentation. So please enter any questions you have into that chat function for discussion then. And I'll be starting with a brief discussion of cell culture methods. These cell culture methods haven't changed significantly since the first methods for animal cell culture were published in the early 1900s. Both then and now, the standard method of primary cell isolation involves cutting tissue into thin slices, digesting that tissue to release the cells, and then seeding those cells onto tissue culture plastic or simple protein coatings consisting of a single protein like collagen, fibronectin, or potentially more complex mixtures like Matrigel. These are the methods for primary cells. The methods for iPSCs and their culture and the differentiation is much the same. In this described cell culture system, cells are exposed to a very artificial environment. We tend to do very little to recreate the natural environment from which cells were isolated or would be found in vivo. These cells respond to the changes in their environment with changes in their gene expression, and then we try to make predictions of in vivo function of cells based on these results in an artificial environment. Part of what's missing here is consideration of the extracellular matrix. While cells are the basic building blocks of life, it's estimated that 70% of human dry weight is extracellular collagen alone. So in this image, you can see the cell at the top, but most of the image here is taken up by the extracellular matrix. And if I take a cross-section of this cell in its native environment, it's very difficult to tell where the cell ends and its extracellular matrix begins unless we label it. What this image makes clear is the components of the extracellular matrix plug directly into cytoskeletal components across the plasma membrane. Here you can see the plasma membrane extending from the top left to the bottom right of the screen as a phospholipid bilayer. The dividing line between intracellular and extracellular is a lot blurrier than sometimes depicted. Cells secrete extracellular matrix, and this matrix in turn interacts directly with cellular components to affect cell behavior. Due to this dynamic reciprocity, it is impossible to probe physiological responses of cells when they are not cultured on an appropriate ECM. While researchers now recognize the importance of microenvironment in determining cell fate, many studies probing its effects can still be overly reductionist or simplistic. As an example, in this study at the top left, researchers created gels of a tunable stiffness by varying the ratio of acrylamide to bisacrylamide, and then coating these gels with simple proteins to get cells to stick. This type of system was used to make conclusions about what the optimal substrate stiffness is for a certain cell type. But there are many other factors involved in how cells sense stiffness. For example, in this bottom paper here, researchers used cookie cutter-like cutouts to force cells to take on a certain morphology. The stiffness, roughness, and surface chemistry of the surfaces the cells were grown on were identical. It's the shape that the cells were forced to conform to alone that was changed. And under these conditions, cells were seen to start down different differentiation pathways. We've been taught to eliminate as many variables as possible to isolate the effects of a specific factor. This becomes more complicated when dealing with extracellular matrix because changing one variable, for example, the compositional ratio of a single protein, in turn affects the stiffness, cell morphology, and the potential for cell-to-cell interactions. In contrast, StemBioSys has taken an approach of trying to be more biomimetic. Rather than trying to attempt to perfectly optimize each individual variable of a substrate, we allow cells to do the work for us. We identify a matrix-producing cell from the tissue of interest, grow it to confluence on tissue culture plastic, and then add a low concentration of ascorbic acid to induce the cells to secrete extracellular matrix. And the cells are then lysed using a non-ionizing detergent, and the resulting matrix is washed and then dried onto the cell culture dish in its intact form. These dishes can then be refrigerated for up to 2 years before use. The end user just needs to rehydrate this extracellular matrix with a saline solution, and then the dishes are ready to be used in a standard workflow for the culture of cells. We found that these cell-derived matrices have distinct architectural, physical, and chemical properties based on the cells that were used to produce the matrix. Over the years, we've demonstrated for a wide variety of applications that these matrices can elicit more biologically relevant phenotypes relative to traditional cell culture methods. Since the initial techniques were developed in the lab of StemBioSys' scientific founder, Dr. Xiaodong Chen, we have completed years of development work leading to highly reproducible processes for the manufacture of matrix in everything from cell stacks to 384 well plates. The process is simple, scalable, and reproducible. You can see the fibrous and intact structure of the matrix in the SEM image at the left, and you can also see it under bright field at this image in the center. But the ability to see it under bright field does not influence the user's ability to be able to image your cells in bright field either. Nor does it interfere with the ability of users to do fluorescent imaging of the cells on the substrate. The matrix also fits into the existing workflows of customers. After that brief rehydration step with saline mentioned, the user can seed cells directly onto the matrix, allowing them to get results that are more reflective of what's happening in vivo. So up to this point, I haven't discussed what particular cell type we're using for matrix production, and that's because we can use a variety of different cell types for this. There are a variety of microenvironments in the body, and while we're being biomimetic, we do not anticipate ever offering hundreds of different types of matrices in order to create a niche for every single cell type. Neither is this a one-size-fits-all approach. We instead recognize that while each cell type has a unique niche, common motifs exist that may make the extracellular matrix appropriate for more than one cell type. Our basic approach is to take matrix-producing cells from the tissue of interest and use those to produce an extracellular matrix. At this point, StemBioSys has neuromatrix that is produced by astrocytes, bone marrow matrix that's suitable for the culture of a wide variety of mesenchymal cells produced by bone marrow MSCs. The hepatomatrix is one that we have under development, and then chondromatrix for the culture of chondrocytes. When we look at the composition, the roughness, and the stiffness and fiber orientation of these matrices produced by the different cell types, we can see predictable differences that translate to changes in the behavior of cells that are cultured on these matrices. The matrix that we will be discussing further today is Matrix Plus, which we have used extensively for the culture of cardiomyocytes. So with that, I'm going to introduce briefly our collaboration with Dr. Heron. And this was founded from the understanding that iPSC-derived cardiomyocytes suffer similar issues of other iPSC-derived cell types in culture. While in theory, iPSCs can produce any cell type in the body, in practice, they produce any cell type in the body-ish. We can make cells that resemble neurons, resemble hepatocytes, resemble cardiomyocytes, but they don't fully recapitulate the phenotype of cells in the adult human. And while there are strategies that can be used to mature cells, they tend to require extended culture periods or more complicated techniques like mechanical or electrical pacing of the cells. So when Dr. Heron's lab first plated iPSC-derived cardiomyocytes onto Matrix Plus, he saw something very different than what was seen on the traditional matrigel substrate. After about 7 days of culture, You could see on the right here how you have rod-shaped elongated cells, many of which are binucleated and have organized sarcomeres that could be seen in a bright field in comparison to the more round egg-shaped fried egg kind of cell look that you see on matrix gel. And this binucleation and organized sarcomeres are characteristic of a mature cardiomyocyte phenotype. And so the enthusiasm from this early work has since led to a years-long collaboration. And STEMbiosis has since developed our own iPSC-derived atrial cardiomyocytes that we produce from the STEMgen iPSCs from REPROCELL. And we recommend the culture of these on Matrix Plus in order to get that cardiomyocyte maturation we've described. These cells we differentiate according to a published protocol using small molecules and then purify them via negative magnetic selection before freezing them for use by customers. And this provides our customers with an untouched and non-genetically modified human cells for their experiments. And so it's these cells that I'm going to turn it over now to Dr. Heron so that he can discuss the results about the chamber specificity of these cells.
Thank you, Anne, for the introduction of the STEMbiosis cell matrices. And I will be introducing data that we have collected at the University of Michigan using STEMbiosis Matrix Plus. So I'll be introducing data on the use of mature chamber-specific human cardiomyocytes, implementing the ECM of Matrix Plus for high throughput cardiotoxicity testing in vitro. By way of introduction, I'm going to begin with a brief review of cardiac chamber-specific functions in vivo with a focus on the electrophysiological function. Then I will move into how we are recapitulating in vivo cardiac function in vitro using human-induced pluripotent stem cell-derived cardiomyocyte assays. Excitingly, the use of these stem cell-derived assays are beginning to reduce, refine, and replace the use of animals for many cardiotoxicity testing reasons that are needed for FDA validation and safety screening. This has been led by international validation efforts by the HESI committee and by the FDA. Then I'll move into how we use human extracellular matrix to induce cardiomyocyte maturation, and also show how the use of chamber-specific cardiomyocytes impacts on drug responses and function that's observed in vitro. Then finally, I'll introduce a comparison of chamber-specific cardiomyocytes from commercial sources. These are cells that are cryopreserved and distributed. One is atrial-specific cardiomyocytes from STEMbiosis, and the other commercial product is a predominantly ventricular cardiomyocyte cell from Fujifilm and Cellular Dynamics. So I've included this to review very briefly and simply the cardiac chamber-specific electrophysiology. On the left is a cartoon of a human heart, and on the right is the corresponding electrical activity that happens with each heartbeat. So with each heartbeat begins at the SA node, and the SA node action potential is shown here. It's a calcium-driven action potential, which means that it has a slow upstroke and a non-stable resting membrane potential. From the SA node, the electrical signal of each heartbeat then travels through the atrium, and in the atrial myocardium, the action potential looks much different from the SA node. It has a very rapid upstroke, and it's sodium driven, and so these cells have distinct ion channel gene expression from the SA node cells. After the signal travels through the atrial muscle and the atrial muscle contract, the electrical signal then slows down and passes through the AV node, or the atrioventricular node, located between the two chambers of the atria and the ventricles. And the AV node action potential looks similar to the SA node action potential and has similar ionic mechanisms. The signal then travels through the AV node to the Purkinje cells, which are the electrical conducting cells on the endocardium of the ventricular chambers. And this is done to ensure speedy delivery of the electrical signal through the ventricles to the working myocardium in the ventricle, shown here, which has yet another distinct action potential profile. And these action potential events, you can see how they contribute to the overall ECG that's recorded, the surface ECG of patients. And importantly, the interval of the EKG that's important for cardiac safety is the QT interval, which is predominantly a reflection of ventricular repolarization. So there are distinct cell types in each chamber of the heart, and they have very specialized functions electrophysiologically and also as they contribute to contraction. And the goal of our in vitro cardiotoxicity electrophysiology screening assays is really to mimic the in vivo heart function that you see on the left. And the best way to do this now is using human-induced pluripotent stem cells, or HIPSCs. And these cells are derived from a patient blood sample or a skin biopsy. And in this case, REPROCELL utilizes mRNA reprogramming to directly reprogram somatic cells to iPSCs. And then once the iPSCs are created, they are pluripotent and they resemble, with remarkably similarity, embryonic stem cells that can be used for differentiation to any cell type of the body. And here I'll be focusing on how these cells are used for cardiac specific differentiation in the production of stem cell derived cardiomyocytes that are used for high throughput screening for drug cardiotoxicity. And as I mentioned in the introduction, this is a very exciting approach because it is beginning to reduce, refine, and replace the use of animals in many cases of cardiotoxicity screening, and has utilization in drug discovery for cardiotoxicity screening. Importantly, FDA guidelines, safety guidelines, are beginning to incorporate in vitro iPSC-derived data for investigative new drug applications. And since this cell type comes from a patient, it also paves the way for future personalized medicine. In the most highly characterized commercial source of human cardiomyocytes that are available from Fujifilm and Cellular Dynamics, these cells contain 3 different types of cardiac cells. One cell type is atrial-like, which you can see here. Another cell type present in these products are nodal-like cells, which are SA node-like cells and have distinct electrophysiological function from the atrial cells. And the predominant phenotype in these cells is a ventricular-like phenotype, where the action potentials have a much larger amplitude and a much longer duration, similar to what is known to occur and be different in different parts of the human heart. But what's not clear is what effect this cellular heterogeneity has on electrophysiological outcomes, and also the implications for cardiotoxicity screening and for use as cardiac research models is unclear. It is certain, though, that having these different cell types present in the same monolayer is a misrepresentation of in vivo cardiac function, where the cells of the atrium are always separately, spatially distinct from the ventricular cells, and you don't find atrial cells mixed into the ventricles of human hearts. In our own research, using I-cell squared cardiomyocytes from Fujifilm, we've done immunostaining for ventricular cells shown in green, and also for atrial cells shown in red. These are stainings that are specific for each cell type, ventricular or atrial. And at higher magnification in panel B, you can really see that here this is creating a monolayer in which ventricular cells, which we know have distinct electrophysiological function, are directly interacting with atrial cardiomyocytes in red, which have a much shorter action potential. And the direct impact of this type of heterocellular connection in the cardiac monolayer is unknown. And the breakdown of the different cell types are shown here, where 8% of the cardiomyocytes are atrial, 44% are ventricular, and then there's even cell types that are positive for both atrial and ventricular cells, which represent about 14% of the total cell population. And again, the research that we're going to do kind of sheds light on the impact of the cellular heterogeneity and the effects on electrophysiological outcomes. I'll also be showing the effect of maturity on outcomes. And again, I just want to remind you that having these different chamber-specific cardiac cell types mixed together is a poor representation of the in vivo cardiac condition. So one solution that we've been able to make for the field is using optimal extracellular matrix to mature the iPSC-CM phenotype of these I-cell squared cardiomyocytes. And here you can see from the same batch of cells on the left, the cells were cultured on Matrigel mouse-derived extracellular matrix, and the cells have a circular phenotype with sarcomeres that are disorganized and radially distributed throughout the cells. And this is what fetal cardiomyocytes look like. On the other hand, the same batch of cardiomyocytes plated and cultured on Matrix Plus human ECM have a much different and much more mature structure. You can see that the cardiomyocytes plated on this matrix have sarcomeres that are in register with one another and run parallel to each other throughout each cell. And we also can see the emergence of binucleated cells in the mature cardiomyocytes, where the ECM is maturing the cardiomyocyte phenotype. This slide is to introduce the very simple method for maturing cardiomyocytes using Celvo Matrix Plus. Here, this is a published method from scientific reports that's also been published in iScience in 2023. And here we take cryopreserved cardiomyocytes. In this case, these are I-cell squared cardiomyocytes. We plate them at a plating density of 75,000 cells per well to create electrically and mechanically connected monolayers. And we let those cells grow for 7 days, allow them to be maintained in culture, and after 7 days, we routinely observe a rod-shaped morphology of the cardiomyocytes, which you can see here with immunostaining for cardiac troponin T in green, which is a cardiac-specific myofilament protein, and on the bottom, staining for alpha-actinin, Z-band protein of the cardiac sarcomere. Also here, you can appreciate the binucleation of the cardiomyocytes that's also known to occur with maturation of cardiomyocytes. This image in the bottom left is a larger field of view showing a greater number of cells plated and grown on Matrix Plus, just to show that the structural maturation of the cardiomyocytes occurs in all of the cardiomyocytes, not just in one or two that we've picked out. The structural maturation can also be observed in phase contrast, just by looking at the rod-shaped structure of each cardiomyocyte plated on matrix plus. And this is a closer analysis of the sarcomere structure of a matured human-induced pluripotent stem cell-derived cardiomyocyte. And here, along this blue line within the cell on the bottom, is a fluorescence intensity plot, which shows the very regular spacing of the sarcomeres, indicated by the spacing of the troponin I band across that line, and a very nice resting sarcomere length of 1.8 microns. In these videos, I'm going to show one video, the video on the left. The first one that should play are single cells that have been thawed. So it's showing how the cells go from being circular shaped when they're thawed to becoming rod shaped over a 7-day period in culture. So here you can see the cardiomyocytes plated. The imaging is roughly every 6 hours. And you can see at the end of the movie that the cardiomyocytes have all become rod-shaped and distinct from fetal phenotype cardiomyocytes that are circular in shape. The next movie that I'm going to show is a time-lapse video of cardiomyocytes plated at a higher density to form continuous monolayers, because in our 96 well plates, the functional unit that we measure is HIPSC CM monolayer. So here you see the cells at a higher density go from being circular in shape to, at the end of the movie, being rod-shaped, even with some evidence of fiber orientation and organization happening across the monolayer. So that was a very big advance for the field and for our laboratory to be able to mature cardiomyocytes in as little as 7 days. Other methods that have been used for maturation include extended periods of time, for example, 100 days of culture, or the use of complicated and sometimes not successful chronic pacing protocols, where the cells have to be paced chronically in culture. The next solution that we've worked on to make cardiotoxicity testing more representative of the in vivo condition is to differentiate stem cells to become chamber-specific cells using small molecules. And to do this, we utilize a published protocol that was published in 2018 in JC Insight, and that we've recently also reproduced in a publication in iScience in 2023. And this protocol is very simple. It utilizes small molecules applied at day 0, and at day 2, and at day 3. And the simple addition of retinoic acid to the protocol at a very specific time point in the differentiation between days 3 and day 6 induce the cardiomyocytes to have a predominantly atrial cardiomyocyte phenotype compared to the ventricular cells. And so this enables us to utilize cardiac monolayers that are chamber specific, where the ventricular cardiomyocytes are made up of exclusively ventricular cells, and the atrial cardiomyocytes are made up of just atrial cells and are more representative of the in vivo condition, where the cells are separated spatially. To do high throughput electrophysiology screening, I've included this slide to show you how that is done. We utilize an optical mapping machine that contains a high resolution, both spatial and temporal resolution camera. And the plate of cells is positioned under that camera, and then a motorized stage moves the plate around under the camera. This machine was first called Cartox in our first prototype, and now it's being sold commercially under the name Nautilus by Curie Bio. And this machine allows us to observe with very high throughput and very robust data collection the spontaneous beating of 96 monolayers at the same time. And utilizing software, we're able to do parallel processing of all wells simultaneously to enable us to generate very robust data sets for very robust cardiotoxicity testing. Some of the parameters that we quantify that I'll be presenting in the data slides are action potential triangulation, which is a standard index of proarrhythmic potential of cardiac cells. And another parameter is the action potential duration 80, which is an in vitro surrogate marker for the QT interval of the surface EKG. And so this is a representative action potential shown in blue that activates each heartbeat. And the parameters that we measure are the action potential duration, which is the amount of time it takes for the return of the action potential to baseline. And the other parameter that we measure is triangulation, which is the amount of time that it takes from this point at action potential duration 40 to this point at action potential duration 80. And so any drug or intervention that prolongs the action potential duration, as I've shown here in red, would be a compound that increases the potential for arrhythmias. So the increased action potential duration corresponds to QT prolongation, and drugs that block specifically potassium channels are known to contribute to action potential prolongation and QT prolongation. This is an example of a positive control compound showing our system's ability to detect arrhythmias. Here we use a drug called E4031, which is a specific hERG potassium channel blocker. And here you can see at a low dose of E4031, this well H7, which was beating with very normal rhythm before adding E4031. And then after adding E4031, we can see a much different rhythm and a much different pattern of the cardiac monolayer, where we can observe APD prolongation and also early after depolarizations shown here, which also can induce arrhythmias. And in the bottom panel, this is a higher dose of E4031, 5 times greater dose. And here again, you can see the E4031 turned what was a normal rhythm into an arrhythmia. So here you can see APD, action potential prolongation, and here the action potential or the membrane potential doesn't return to baseline, and this is what occurs during TDP arrhythmia. The TDP stands for Torsade de Pointes arrhythmia. So here, this is showing and validating how we make chamber-specific cardiomyocytes. One very good myofilament protein that is an excellent molecular marker to discriminate between atrial and ventricular cells is the myosin heavy chain that's expressed. It's well known in adult human atria do not express beta myosin heavy chain, but they do express the other myosin heavy chain called alpha myosin heavy chain. Here in this Western blot, we can see that the ventricular cells that we make do indeed express high levels of beta myosin heavy chain, while the atrial cardiomyocytes do not express significant levels of beta myosin heavy chain. And here we've done experiments since we're able to generate atrial specific cells as well as ventricular specific cells. We also did some experiments where we mixed the two cell types together to create a more heterocellular monolayer. And here you can see an intermediate expression level of the beta myosin heavy chain when we do a co-culture, mixing the two chamber specific cell types together in vitro. In panel C, you can see the different chamber-specific electrophysiological functions that we observe in these in vitro assays, and they're very similar to what's well known to be different about the in vivo function of the atria and the ventricles. So here, this is an action potential recorded in the laboratory, The atrial action potential in brown is much shorter in duration and amplitude than the ventricular action potential, which is shown in blue. Remarkably, in the co-culture situation where we mixed atrial and ventricular cardiomyocytes together, the action potential profile is right in the middle between the two extremes of atrial and ventricular cardiomyocytes, indicating that the co-culture of the two cell types has significant impact on the overall function of the cardiac monolayer. And this is the quantification of that data, showing that, again, in brown, the atrial action potential duration is much shorter than the ventricular action potential, and the co-culture values are intermediate in between the two extremes. And the same holds true in panel E for the action potential triangulation, which is determined by the action potential duration. You can see, again, segregation of the function based on the cell type in the cardiac monolayers. So this data indicates that we can generate chamber-specific cells that have chamber-specific myofilament expression, and from the action potentials, we can infer also distinct ionic current and ionic channel, membrane channel expression. So another question is, what is the impact of this on drug responsiveness? I just want to point out also that when we do higher temporal resolution imaging of action potentials in the atrial monolayers, we also see the characteristic notch and plateau phase of the action potential shown here, which is reminiscent of what's known to occur when you record human atrial action potentials from human atrial tissue, where there's a spike and dome or a notch and a plateau phase. So here we have tested the response of atrial specific cells shown in brown in panel A, or ventricular specific cells shown in blue in panel B, to the same medication. In this case, we tested a medication called vernaculant. This drug is approved in Europe for atrial fibrillation conversion to sinus rhythm. However, in the United States, the drug is still at an investigational new drug status by the FDA. And this drug is known to be a multi-channel blocker that also binds to atrial-specific ion channels. So here we can see a dose response to vernaculant for immature cells cultured on Matrigel compared to mature cells cultured on Matrix Plus extracellular matrix. And you can see greater conduction velocity in the mature cells plated on Matrix Plus ECM, and also a difference in sensitivity to the effects of vernaculant to slow the conduction velocity of the cardiac action potential. In the ventricular cells, we can see a similar effect where the vernaculant also slows conduction velocity, and this is likely due to its effects to block sodium currents. If we look at the calcium transient duration 80 in the bottom, here we can start to see very discrepant effects between atrial cells in panel C and ventricular cells shown in panel D. Notably, we can see that the calcium transient duration 80 is not significantly impacted by vernaculant. However, in the case of the ventricular monolayers, we can see a very robust and a very dose-dependent effect of vernaculant to prolong the calcium transient duration of ventricular cells. Here, this is plotted the early repolarization, or the early decay of calcium in atrial monolayers, again in brown on the left, compared to the response in ventricular cells on the right. And here again, we can see distinct chamber-specific responses to the same medication at the same doses, where the atrial myocytes are much less sensitive to vernaculant compared to the ventricular cardiomyocytes, which have a very dose-dependent and much greater magnitude of prolongation of the calcium transient duration. So the use of chamber-specific myocytes does have implications for cardiac safety screening and certainly has implications for the development and efficacy testing of chamber-specific antiarrhythmic medications. Here, when we look at the calcium transient duration triangulation, we can see the opposite effects very clearly of vernaculant, where in atrial monolayers, the vernaculant is actually antiarrhythmic, whereas in the case of the ventricular cells, the vernaculant appears to be proarrhythmic. And this is likely the reason that vernaculant is not approved for use in the United States. Next, I'm going to present a comparison of chamber-specific cells obtained from two different commercial sources. In one case, I'll be showing data of ventricular cardiomyocytes from Fujifilm and Cellular Dynamics compared to the function of atrial-specific cardiomyocytes from StemBioSys. Some differences between the two products. One is that the Fujifilm product contains ventricular myocytes at a higher proportion than atrial myocytes, which are also a higher proportion than nodal cardiomyocytes. However, the StemBioSys product contains exclusively atrial cardiomyocytes. Also, the Fujifilm product is genetically modified to promote antibiotic selection of cardiomyocytes. On the other hand, the StemBioSys cells are made from mRNA reprogrammed stem cells available from REPROCELL, and there's no genetic modification. And for purification of the atrial cardiomyocytes, magnetic activated cell sorting is used, and this is a clinically approved method for the preparation of clinically used cells for therapies. This is a trace, representative trace of the calcium transients of the Fujifilm cells shown by the black trace, where the beat rate is much slower and also of greater amplitude than the atrial cardiomyocytes shown in purple, which have a much shorter duration of the calcium transient and also a much faster beat rate than the predominantly ventricular cardiomyocytes. Here I've superimposed one activation from each monolayer cell type, just to show the obvious differences in the amplitude of the calcium transient between the ventricular and the atrial cardiomyocytes, and also the duration of the electrophysiological event being much shorter, as expected in the atrial cardiomyocytes. This is a quantification of the baseline comparison between I-cell squared cardiomyocytes and StemBioSys salvo-atrial cardiomyocytes, showing that the atrial myocytes have a much faster beat rate, a lower calcium transient amplitude, which would indicate also smaller contraction amplitude, using the calcium transient as a surrogate marker for contraction. And regarding the calcium transient duration, the atrial cells have a much shorter calcium transient duration, likely due to the increased beat rate of the atrial cardiomyocytes. And next, I think there will be required validation using medications with known effects, with known chamber-specific effects, to see how that plays out in the future. We've also done some preliminary medication testing here using isoproterenol. Isoproterenol is the classical positive inotrope of the heart, which mimics the effects of exercise. So isoproterenol, when applied to the human cardiac monolayers, should increase the beat rate, as happens with exercise, and also an increase in the contraction amplitude. And so what we can see, if we look at the effect of isoproterenol to increase the beat rate or the heart rate, the ventricular cells here are shown in black and the atrial are shown in purple. We can see that in both cases, there was an increase in the spontaneous beat rate caused by isoproterenol. When we look in the middle panel, the middle data, looking at the calcium transient amplitude, this is an index of the inotropic state of the monolayers. And in the ventricular cells, we see some monolayers with an increased inotropic state and others that decrease. But in the atrial cells here, we can see predominantly a positive inotropic response. And when we look at the response to isoproterenol to hasten the duration of the calcium transient, we can see that in both cases that this lusotropic effect, positive lusotropic effect of isoproterenol is apparent. We've done some gene expression analysis by qPCR to look at the expression here of atrial specific genes, what are known to be atrial specific genes. And we can see that compared to the ventricular cells, the stem biosys atrial myocytes have much more expression of MYH6, which is the alpha myosin heavy chain gene of the cardiac myofilaments. And also the atrial myocytes have a greater relative expression of the NPPA gene, which is the gene that encodes for atrial natriuretic peptide. We also have looked for ventricular specific gene expression by qPCR. Here, probing for myosin heavy chain 7 gene expression. This is the beta myosin heavy chain. We can see much greater expression in the ventricular cells. Looking at myosin light chain 2, which is another ventricular specific protein and genetic marker, we can see much more robust expression of myosin light chain 2 in the ventricular cells. And looking at HEY2, which is a ventricular specific transcription factor, we can see that there's almost no detectable expression in the atrial cells, but very robust expression in the ventricular cardiomyocytes. This is an example of another experiment that we performed recently, where we used seahorse plates that are available from Agilent. And these plates are specially designed to measure metabolic function to get an indication of the mitochondrial function of cells cultured in the seahorse plate. And this plate was coated at Stem Biosys with Matrix Plus and then shipped to my laboratory at the University of Michigan. We then proceeded to plate cells, and we divided the plates into 4 different quadrants. On the left, these are atrial cardiomyocytes, shown in red. All of these wells were made up of atrial cells. And on the other hand, on the right half of the plate, these were all ventricular predominant cardiomyocytes from Fujifilm. So we plated these to form monolayers on the dish, and then we used an ischemic mimetic media that we call heart attack in a dish media. And the bottom half of both the atrial and the ventricular cells were cultured in this ischemic heart attack media for a total of 7 days. The difference between the two medias, the control media contains normal levels of glucose, whereas the ischemic media is a media that has 0 glucose, but abundant lactic acid to mimic the conditions that occur in the heart during myocardial ischemia, which occurs typically secondary to an occluded artery or coronary artery disease. So we took images of the cells from each condition after the 7-day treatment with control media or with the heart attack media. On the left are the atrial cardiomyocytes. The top left, these are atrial cardiomyocytes maintained in control media. You can see they have a nice rod-shaped morphology and have very nice monolayers. But whenever they're treated with the ischemic media, you can see that the cells become sick and really do not look good. Similarly, on the ventricular cells in the control media, they look good. The cells are rod-shaped and function normally. However, after 7 days of treatment with the ischemic heart attack media, the ventricular myocytes also begin to look very sick. And in this assay, using the seahorse plate, we actually took an opportunity to multiplex the assay, where we combined electrophysiological measurements and contractility together with the seahorse XF plate to also measure mitochondrial function in the same plate with the same wells. And so here, again, in this experiment, you can see the reproducibility of the differences in contractility between atrial and ventricular cells, in the duration of the calcium transient between atrial and ventricular cells, and also the profound difference in the beat rate between the atrial and ventricular cardiomyocyte monolayers. Here, this shows the response of those same parameters for each cell type to the ischemic media. Looking at contractility, you can see that in both the atrial as well as the ventricular cells, there's a significant reduction in the calcium transient amplitude, which indicates a reduction of contractility. Looking at the calcium transient duration, we can see significant prolongation of the calcium transient duration in the atrial cells treated with the ischemic media. And looking at the heart rate or the beat rate here, you can see that in the atrial cells, the ischemic media causes a significant reduction in the beat rate, but in the ventricular cells, there was no effect. Again, showing the differences in chamber-specific cells to the same insult here in ischemic media challenge. We also performed on the same plate as part of the multiplexing the seahorse measurements, measuring the oxygen consumption rate, which is an indication of mitochondrial respiration. And here you can see the effect of ischemia on the atrial cardiomyocytes. So the control curve is shown in blue, and the ischemic atrial is shown in red. You can see a major reduction in the metabolic function of the cardiomyocytes induced by the ischemic media. Similarly, with the ventricular cells, we saw a reduction in the oxygen consumption rate, the maximal oxygen consumption rate between control and ischemic ventricular cardiomyocytes. This is the same data normalized, showing similar trend with normalization, where the ischemic media in both the atrial and ventricular cells have profound effects to reduce the oxygen consumption rate and the mitochondrial respiration function. This is that data quantified for basal respiration on the left, showing the effect of chamber-specific cells on the metabolic state of the monolayers. And also looking in the center at the maximal respiratory capacity, you can see that there's a major reduction of that parameter and mitochondrial function with the ischemic media. And similarly, the spare metabolic capacity is also reduced in both atrial and ventricular cells in the presence of the ischemic media conditions that simulate heart failure. This is another measurement from the seahorse plate, measuring the extracellular acidification rate, which is an index of glycolysis and the use of glycolysis for making ATP. And here you can see, again, differences between control media and ischemic media. Also, you can see differences between atrial cardiomyocytes in blue at baseline and ventricular cardiomyocytes in green in their use of glycolysis. So in summary, it's really remarkable. It's helped our research tremendously, and I think has made for better cardiotoxicity assays, is the rapid structural maturation of cardiomyocytes using human ECM, Selvo Matrix Plus from STEM Biosys. I've shown the important impact of using chamber-specific cardiomyocytes on pro-arrhythmia screening and in mimicking the in vivo condition in vitro for better cardiotoxicity testing. I've shown the use of chamber-specific cells and the impact on metabolic function in response to ischemic conditions. And importantly, now there's a very good commercial product of human atrial-specific cardiomyocytes that are available from Stem Biosys that are created from a cell line from REPROCELL. And with that, thank you for your attention.